Okay. So we're going to take one of these pyruvates and we're going to show you what happens to it the rest of the way. Understand, both pyruvates would do the same thing. I'm not going to keep following both pyruvates. We'll follow one pyruvate at a time, and just understand that there are two pyruvates for every sugar that go through this process. So let's go to the next step. Some sources call it the intermediate step. Some sources call it the link step. I'm trying to remember what the Pogil called it. It might have called it the oxidation pyruvate. Different sources, they use different names. Some don't even consider this a separate step. It just depends on what source you listen to. This step really is preparing the molecule to be able to access the mitochondria. This step is about getting the molecule ready to enter the mitochondria and complete its breakdown. So we're going to pick up with the pyruvate. Which again is 3 carbons. And it's a very simple step. We're going to convert this pyruvate into a 2-carbon molecule in the process. So we're going to oxidize pyruvate into this 2-carbon molecule. And I'll add some more detail to that. And then we're going to convert that into a slightly different 2-carbon molecule. And that's going to involve an arrow there. And that's going to involve a little bit more here. That's going to involve a little bit more here. Okay. So that's the basic diagram or basic schematic here. All right, so I'll let you catch up on that. I just got to grab one thing from my desk real quick. Let me draw that out. I've disappeared. I'm coming back here in just a second. I'm just checking the language that the Pogil used so that we're being consistent. Okay. So I just was double checking. In the Pogil, they do call it the link reaction. So intermediate step, link reaction. Similar idea. So what's going to happen is we're going to oxidize the pyruvate. And this time it's going to cause us to release one of the carbons. We're going to destabilize the molecule to the point that it can no longer hang on to one of the carbons. And one of those carbons is going to be released, and it's going to be released in the form of carbon dioxide. So there's going to be some CO2 released. And because we're oxidizing it, again, we're going to release a pair of electrons, and we're going to release some hydrogen. Only this time, there's not quite enough energy to produce any more ATP. So the energy that's with the electrons is going to, again, get reduced. If I oxidize the pyruvate, I've got to reduce something. I'm going to reduce, again, a molecule of NAD plus into NADH. It's picking up the electrons and the hydrogens. And I'm going to produce something called acetic acid. Two carbon molecule called acetic acid. However, before that acetic acid can get all the way into the mitochondria, I need to provide it sort of with a pass, a chemical pass, I guess you would say. There's a coenzyme. Remember, coenzymes are sometimes needed by enzymes in order to function. There's something called coenzyme A that we're going to add into this into this molecule, okay? And when I add the coenzyme A to the acetic acid, I create a molecule called acetyl-CoA, which is basically acetic acid with coenzyme A attached. So we call it acetyl-CoA, C-O-capital-A, acetyl-CoA. Acetyl-CoA now has the chemical structure necessary to pass through the outer membrane, the inner membrane space, and through the cristae all the way into the matrix. This is going to give it entrance all the way into the matrix. So it allows the molecule to enter the matrix of the mitochondria. Remember, the matrix is the inner space that has got a dense fluid in it. It's where the Krebs cycle is going to take place. So if we do an accounting here, in this case, carbon dioxide, we have, so I'm going to do actually a little bit different here. We'll put per pyruvate. So how much do we make for each pyruvate? And I'm going to put per glucose. Is that making sense? It will in a second. So for per pyruvate, I'm releasing 1 carbon dioxide. But per glucose, there's 2 pyruvates. So since there's 2 pyruvates, 2 times 1 is 2. Overall, for every glucose, I'm producing 2 CO2s. So of the 6 original carbons, the link reaction is going to release 2 of the 6 carbons into the atmosphere. Remember, matter is neither created nor destroyed, but it can be changed from one form to another. In this case, NADH, I have 1 NADH per pyruvate, total of 2 per glucose. In this case, FADH2, the other carrier, I have 0, so I'm going to have 0. In terms of ATP, I have 0 and 0. There's not enough energy being removed to produce any ATP at this point. And water, I am not producing any water at this point. So I've just broke the molecule. All I've done in this step is broke the molecule down in just a little bit finer point. So inputs are pyruvate. What's coming out of that pyruvate? A couple of carbon dioxides and a couple of NADHs. Nothing hugely significant outside of you've prepped the molecule so it can go into the mitochondria. This is a necessary step so you can enter the mitochondria so you can finish harvesting the energy. This is the necessary step to prep the molecule to enter the mitochondria so you're able to harvest the energy from it. So first 2 steps, we got a couple carbon dioxides, we got about 4 NADHs, we got a couple ATPs. Most of the energy is still part of the original sugar molecule. So the next step is to finish getting the rest of the energy out of the sugar. Again, we have to do this in slow, small steps so we don't release too much energy too quickly. Questions at this step? Okay. I realize it's a little difficult to ask anything yet because you're still trying to figure it all out. It will all connect when we finally get through all 4 steps. All right, so now that we have acetyl-CoA, we have moved into the mitochondria. We've moved into the mitochondria. So we are now inside the organelle. We are all the way in the interior. We are in the matrix, the dense fluid that sits inside the inner membrane. And that is where we're going to have what's called the Krebs cycle or citric acid cycle. I just do a little mini drawing of the mitochondria, we are right here. Right here in the matrix. Innermost part of the mitochondria is where this is occurring. It is like the Calvin cycle, a cycle. It is a cycle that repeats itself over and over and over again. All right. Again, there are a couple dozen enzymes. There's about 8 or 10 different intermediate products. We're not going to go into all the intermediate products. We're not going to go into naming all the enzymes. If you end up going into some type of nutritional field or dietary field, you might get into that stuff in your junior, senior year of college. So anyway, I'll do the schematic again for this, and we'll try to explain what's happening with the Krebs cycle. I probably will not go into the final step until we'll see where we're at and how people are feeling. I probably won't go into the final step till Friday. Just to give you time to process. But we'll see. It might be better just to go through the whole thing and we'll find out. So just got to think for a second as to how many boxes I want to use. Right? I don't want to make this too complicated. Let's go there. Again, you would not be expected to redraw this in any way, shape, or form. You might be given a diagram that looks similar to this, and you should be able to interpret it, but you would not be asked to reproduce this. All right, that's going to be there. We're sort of setting it up first, and then we'll walk through it. Okay. Also going to note that a lot of this requires enough room very well. This whole cycle is driven by water. That's what I'm trying to illustrate. Each step involves the input of water. Remember the process hydrolysis. Hydrolysis is the main way that we break down molecules. You add water to break the bonds. So this whole cycle is a hydrolytic cycle. It's all driven by water. And so what's happening in this cycle? So let's try to add some of the details. I'm not going to, again, get stuck up too heavily on names and things of that sort. So you have a 4-carbon molecule right here that is the beginning of the cycle. It's the carrier molecule. If you remember in the Calvin cycle, this was something called rubisco. Robisco was what picked up the carbon dioxide and fixed it. It did the carbon fixation. But at the end, Robisco was recreated. This is something called oxyl acetate. I'm not going to worry about the name, but this 4-carbon molecule is going to pick up the acetyl-CoA, take it around the Krebs cycle, and then you'll end up with the 4-carbon molecule being regenerated. So it's reused. So this 4-carbon molecule is going to pick up this 2-carbon acetyl-CoA. This is what came out of the link step. So this is the result of the link step, this acetyl-CoA. So when you combine the 4-carbon carrier molecule, this 4-carbon carrier, with the 2-carbon acetyl-CoA, that is going to create, of course, a new molecule that's got 6 carbons. 2 plus 4 is 6. And so we have this 6-carbon molecule, which is called citric acid. I only give you the name this time because that's where the name citric acid cycle comes from. So it is the citric acid cycle, or also the Krebs cycle after the dude who figured it out. So the 4-carbon molecule right here picks up the 2-carbon molecule that comes from the link step. Let's note that that's coming from the link step. And that forms this temporary 6-carbon molecule called citric acid. So now what we're going to do is we're just going to keep oxidizing and reducing so we can break apart the rest of the sugar. This is what's left of the sugar. This is what's left of the sugar. This is acetyl-CoA. And so what we're going to do is we're going to pull apart the rest of that acetyl-CoA and extract the energy. And when we're done, we're going to recreate the original 4-carbon molecule so it can do it all over again. So there's several points in the reaction where we add water and oxidize the molecule. So the first step, we're going to oxidize this 6-carbon citric acid into a 5-carbon molecule. Again, I'm not going to get hung up on the name, which means we're going to lose some CO2. So we lost a carbon. In the process, we are also going to be releasing electrons and hydrogen, which is going to allow us to reduce NAD plus into NADH. So we've oxidized the 6-carbon molecule, we reduced the NAD, and we created a new 5-carbon molecule. Oxidized, reduced, oxidized. Can't see where I'm pointing. Oxidize this, and we reduced to here. Next step, we're going to do very similar. We're going to take a 5-carbon molecule. We're going to take this 5-carbon molecule. We're going to add water, and we are going to oxidize it into a 4-carbon molecule. It's a different 4-carbon molecule than the final product, but we're going to make a 4-carbon molecule. Again, we're going to release some CO2. Some CO2 is going to be released. And again, we are going to provide some electrons and hydrogen so that we can form NADH. And this time, because we've now broken down, there's 2 carbons are now broken down. 2 carbons, there's one of them. Turn into CO2 right here. And here's the other CO2 here. We've broke off the final 2 carbons, and it provides us with just enough energy to make 1 ATP. We can make 1 more ATP. Now, this 4-carbon molecule here is not quite the same as this one here, so we've got to do a final step. We're going to do 1 more step, add a little bit more water. We're going to release a few more pairs of electrons, a couple more pairs of electrons, a couple more hydrogen ions, and we're going to, again, that's being oxidized. We're oxidizing this 4-carbon molecule, and we're going to reduce NAD to NADH. And this final pair of electrons that is being released right here has a little less energy than normal. There's not much left. We're at the end of the tank. There's not quite enough energy left to make more NADH. It takes a little bit more energy to make NADH. So we got to use a different carrier molecule because there's less energy available. So this is the one time that we bring in FADH, because FADH is a carrier molecule that doesn't need as much energy. And we're going to convert FADH with this final pair of electrons into FADH2. Convert that into FADH2. So now we got a big mess. Not really. So again, no way, shape, or form are you going to have to redraw that. But if you're given a similar diagram, hopefully a little bit neater, could you interpret basically what's going on? And from that, what are the inputs and the outputs? Again, I'm going to do per pyruvate, and I'm going to do per glucose. So I just put PY for pyruvate, GL for glucose. So if we're looking at CO2, I have 1 CO2, 2 CO2s. So for every turn of the Krebs cycle, which is 1 pyruvate, for every turn, it's going to be 1 turn. For every glucose, it's going to be 2 turns. There's going to be 2 CO2s produced. But for every glucose molecule, therefore, there'll be a total of 4 produced. So that means we lost 2 CO2s in the link step. We now lost 4 CO2s in total through the Krebs cycle, which means we've lost all 6 carbons that used to be part of the original sugar. If I go with NADH, I have 1, 2, 3. I have 3 NADHs that are being produced here. So for every glucose, because it's 2 turns for every glucose, that would be a total of 6. The other carrier molecule, which so far we've had none of, we finally have it used once per turn, so a total of 2 times per glucose. ATP, we have 1 per turn or 2 per glucose. Whoops. And to this point, water, we still have none. So Krebs cycle. What's going in is the acetyl-CoA. Just realize the acetyl-CoA is the remnants of what's left of the sugar. The acetyl-CoA is the remnants of what's left of the original sugar. So we're still working on the sugar. What's coming out of this? In total, 4 carbon dioxides and a bunch of electron carriers, 6 NADHs and 2 FADH2s. Keep thinking of those NADHs and FADH2s like a shuttle bus. They're currently holding the energy that used to be in the bonds of the sugar. The energy that used to be in the bonds of the sugar are now in the bonds of those carrier molecules. They're in the bonds of the NADH and the FADH2. It's a fairly stable way to hold on to the energy in small, safe doses. Now we have to harvest that energy and put it into a usable form, particularly ATP. We have to harvest that and put it into the form of ATP. And we've produced, at this stage, 2 more ATP. So if you go through the first 3 steps, glycolysis, the link step, and the Krebs cycle, you've got 6 CO2s. You've got a whole bunch of NADH and FADH2s, which are holding on to the electrons and the energy. And you've got a whole whopping 4 ATP. Still not enough to support something as complex as ourselves. So the final step what's often referred to as the electron transport chain is where we're going to get the bang for the buck. This is where we're going to take all that NADH and FADH2 and convert it into usable energy in the form of ATP. Whew. Questions at this point? Again, just unmute yourself because I may not necessarily be able to see all of you. I can see most of you. So that's about where the POGIL took you, was to that point. We have not gotten to the payoff step, the final step, the oxidative phosphorylation, the electron transport chain, pretty much the same thing. That's where the payoff's going to be. All right. So let's do this for a moment. What is it? It's 8:21. I'm going to need, I'm just calculating what's the best way to use the time. Let's do this. Let's take just a walk away from the screen and scream and take a break, get a drink, use the bathroom, do something, clear your head. So let's come back at like 8:26. That's 4 minutes from now. And we'll pick up from there. So just take a quick mental break for just a second, and I'll be back in about 4 minutes. All right, if you're out there, come back on camera, please. Yep, I lost the whiteboard. What's going on here? All right, for some reason the whiteboard disappeared. It's blocked. Okay. Anyway, we'll get that figured out in a second. So, anything at this point? Okay, so what I'm going to do is I'm going to pause the story here. I'm going to leave you hanging. All right, give you something to look forward to on Friday when we finish the story. So right now, where's all the energy? All the energy is in those NADH and FADH2 molecules. They are all the molecules that are temporarily holding on to the electrons and hydrogen. So the final step is we're going to take all those NADHs and FADH2s, we're going to oxidize them in order to release the energy and convert it into ATP. And that's where it will happen in the final step. So I'm going to talk more about that final step on Friday, just so we can have a little bit of a change of pace here. So what I want to do is, let me come out of sharing this so I can share something else. What I want to do is talk about one of the labs we normally would be doing in class, and we're going to sort of do a walkthrough of the lab, and then you're going to still do the data analysis of the lab. So let me change screens here 1 second. All right. So I'm going to move back to the lab area. All right, so where I need you to go, if you go, let me just share my screen a second. If you go into Google Classroom, you will see the most recent assignments, CE11 and CE12, which are both due before you come back to class on Friday. So what we're going to be looking at right now is CE11. I'll mention it while we're there. CE12 is the homework for Friday. That's going to go through the final step. This is going to have you explore, well, how do we get all the energy? Again, I encourage you, as it says, I encourage you to try to do it with your team. I think you'll get more out of it if you work together on it. I'm not requiring that. I'm just encouraging that you work with your team on it before you come to class on Friday. What I want to focus on right now is CE11. There are several documents. The one we want is this first one right here for right now. I will just point out, this is the actual data in a spreadsheet. So if you end up wanting to do some of the data analysis and some of the graphing on a spreadsheet, all the data is already in the spreadsheet right here on the second one where it says Google Sheets. This PDF is the actual original directions to the lab. You may need to reference that to answer some of the questions, but I'm going to walk you through how the lab normally is done. So I'm going to need you to open up that first document that looks like this, okay? And we are going to go from there. I'm going to move back here to the lab area. All right, so what you're seeing right here, as it says, is below is the data collected by SCHS AP biology students in 2019. Again, last year we did the course in a slightly different sequence, and we were out of school by the time we got to this lab. So we did not get to run it in person last year either. But this is actual student data. This is real student data that was taken by students here at St. Clair High School. And this is the data that you're going to use to respond to the results of the lab. You'll be asked to come back to the independent variable, the dependent variables, the research questions, the null and alternative hypotheses. You'll be asked to calculate the rate. This explains how to calculate the rate of the reaction. You'll be asked to create two different graphs. Up here it describes what the two different graphs should be. Again, if you'd rather do those on a spreadsheet and attach them, that's fine. And then there are some post-analysis questions, a little bit about the design of the lab, which I'm going to go through in just a moment. And there is some questions about the results of the lab. Right, so this is the document you will be completing before coming back to class on Friday. Again, this one, the Google Sheets, this has the raw data. So if you want to just copy and paste so you can create graphs, that's cool. It's right here. You're after the class averages in most cases. We had 8 different teams doing the lab. And these are the original directions. Okay, so what we're setting up is what's called a respirometer. This picture right here is an example of the type of respirometers that we set up. A respirometer is a device for measuring respiration. Capiche? And you can get ones now that hook to the computer and are computer run, and they're really nice. But unfortunately, they're a little beyond our budget. They're not outrageous, but they're still a little higher. They're about $200 a device. We would need about 8 of them, so they're $1,600. Actually, more than that because we need 3 per team. But anyway, we're going to use a little bit more of a simplistic device. The problem with the more simple device is it's more prone to error, though I think we did a pretty good job collecting the data last time we did it. So what we're looking at in this investigation, let me stop sharing here. What we're looking at in this investigation is we're looking at creating a respirometer to measure the rate of respiration. We're going to measure the rate of respiration. So let me show you the setup first, and then I'll show you the math and science behind it. So what we're doing, and I don't know how I'm going to get this to show up. We'll see. Can you see this bag of peas? So we would be working with peas. All right. This is just the standard dry pea that you can buy and plant and grow pea plants with. Or you can soak them in water and cook them and eat them. Normally we'd have a set of peas that have been sitting for 3 days and are germinating. The root is growing out of the pea, and the pea is in the process of germinating and growing. I didn't do that because I knew we weren't going to be here to set it up ourselves. So we would normally have some peas who are germinating. They would be much more plump. They would be full of water because they soaked in water for a couple days. They would have a root growing out of them. And then we would have the dormant peas is another group. Okay. And so we've got what we call respirating peas and non-respirating peas. And we are, in this investigation, going to test two things. We are going to test, do the germinating peas. We're going to compare the germinating peas versus the non-germinating peas. So that's one of our independent variables. Do germinating peas use more oxygen? Do germinating peas use less oxygen? Do germinating peas use the same amount of oxygen as non-germinating peas? And if there is a difference, why is there a difference? And the other factor that we change in this investigation is the temperature. So you're going to set, normally you would set up the experiment once using warm water, right around 37 degrees Celsius, which would make the environmental temperature 37 degrees Celsius, which is around body temp. And then the other time you would run it again, but this time you would set it up with the environmental water temperature being around 10 degrees Celsius, which is a fairly cold temperature, just slightly above freezing. So in this investigation, we're looking at two variables, germinating versus non-germinating, and we're looking at warm temperature versus cool temperature. Capiche? So we have a respirometer. This is a jar. I don't know how well you can see. In this jar, sometimes we just use a beaker. This is the fancy one. In this jar, we have a piece of foam, and I have a device which is holding 3 test tubes. As I'm pulling one test tube out, can we see this okay? Okay, so it's got 3 test tubes, which are just in a plastic lid. There's a foam piece, just to sort of help keep the test tubes from touching each other. And it goes into this jar. Okay, and so the jar is how I'm going to create the environment. First time through, I might put, like I said, water that's about 37 degrees in here. So the tubes would be sitting in a 37-degree bath. That would be the environmental temperature. With me? And then the next time I would empty it out and I would put in water with ice and get it down to 10 degrees. And I would let this equilibrate to about 10 degrees and I would run the experiment again with 10 degrees. Still following? Yes, no? All right. So let me show you how I would set up each tube. And again, this is in the directions. So in each tube, what I need to do is to set up a set as an experimental group. And I'm going to set up a total of 3 tubes per container. All right. Should I show you the math yet or not? So one of the ways I can measure respiration is by how much oxygen is being consumed. Because during respiration, oxygen is consumed. And so this device is going to allow me to measure how much oxygen is being consumed and at what rate the oxygen is being consumed. So I'm going to set up a test tube. And one of the issues is I want to measure the oxygen, but I don't want to measure the CO2. Because yes, I can measure oxygen being consumed, but as oxygen is being consumed, carbon dioxide is also being released. Making sense? So I want to measure the oxygen, but I don't really want to measure the carbon dioxide. So I've got to have a way to negate the carbon dioxide that's going to be released. Because if respiration is happening, oxygen is being consumed, but CO2 is being released. So I have to have a way to counteract the CO2. So what I have to do is I need to take some cotton. I'm just going to do a very primitive setup. I take a wad of cotton, and I would take what's called an absorbent cotton, a cotton that is designed to absorb material. Most cotton is absorbent. And I would place this down into the bottom of the tube. Yeah, here we go. Come on. I would put that down to the bottom of the tube. And then I would add a chemical called KOH. I'll explain the KOH in a little bit, but there's going to be a chemical that goes in here, KOH. It's a pretty reactive chemical. You have to be careful. You don't want to get it on your skin. You don't want to breathe it in. But the KOH, what the KOH is going to do is it's going to react with the carbon dioxide and pull the carbon dioxide out of the air. That's the role of the KOH. It takes the CO2 and it locks it up into something called potassium carbonate. So it pulls the CO2 out of the air, so it's no longer in the air. So it's not affecting my measurement of gases. Does that go anywhere? So I'm going to put in the KOH. The purpose of the KOH is to absorb, in a sense, absorb the CO2. So I'm not measuring the CO2. I'm only measuring the oxygen. Yes or no? The problem is the KOH is toxic to the seeds. So then I take another cotton, which you can't tell on the screen. I take another type of cotton called non-absorbent cotton. This cotton doesn't absorb stuff, so it blocks the CO2 from making contact with the peas, or the KOH from making contact with the peas. Am I making sense? So I'm going to add a non-absorbent cotton, which I know on the screen doesn't look any different. Like so. So I've got the KOH down here in the bottom. And I have just enough to make the cotton damp. I don't have it so it's running liquid. And then I put the non-absorbent on top to protect the peas from being damaged. And then I count out, I forget the exact number, it's in the directions. I think it's 25 germinating peas. All right. So let's assume these were 25 germinating peas here. These are not germinating peas. But if they were germinating peas, I would put them in. Whoops. Dropped a couple of them. I would put them into the tube, and then I would seal the tube with this device here. All right. What I have here is a rubber stopper, and I don't think you're going to be able to see, and it's been sealed with this glass tube, and this glass tube is marked off in millimeters. And I'm not sure you can see that where you're at. All right. And so that's one of my setups. I have the KOH, which is going to absorb the CO2. I have the seeds, and then I have this device here. This is how I'm going to measure the gas, which I'll explain in a second. So that would be one of the setups. Now, if these were germinating seeds, they're going to probably go all the way up to here because they're like 3 times the size. Yes or no? So I'm going to measure that and fill that up. I want to keep the volume the same. So I'm going to step away 1 second. So what I do is I put some water into a graduated cylinder. So maybe I put it at like 20. And what I do is I add my peas. I would have added my peas before I put them in the tube. And what that's going to do is it's going to displace the water. It's going to cause the water to rise in the tube. Yes, no? And that's going to tell me that 25 germinating peas displaces the water, say 50 units. So when I set up the next tube with the dry peas, I'm going to fill this with dry peas until I get the same volume. And that'll tell me how many dry peas to put in the next one. And then I have some pea-sized plastic beads to make up the difference. So this one would have 25 germinating seeds. The next tube would have 25 seeds with some plastic beads. So I don't want to change the number of, I still want 25 seeds and 25 seeds, but I got to have the volume the same. So the plastic beads make up for the extra volume. Is that going anywhere? And then I create one tube that has nothing but plastic beads. That's sort of my control group, just in case there's any minor changes in the surrounding atmosphere. Capiche? Once I've got all three set up, because I don't have time to set up the whole thing, unfortunately, right now, but I'm going to show you a video that will help. So once I've got it all set up, I've got all three tubes in. They all three have these pipettes coming off of them. Can you get the sense? What I'm going to do is put a drop of colored liquid at the end of each tube. I'm going to take a syringe and put a drop of colored liquid. As the peas consume the oxygen, that's going to pull the gas out of the tube. And so as the peas consume the oxygen, that colored drop of liquid is going to travel down the tube as the air gets used up. And that's going to give me a way to measure how much air is being used. Does that make any sense? And so the measurements you're looking at is how far did the dye molecule move to indicate how much air was being consumed by what's in the tube. Does that go anywhere? If I did not have the KOH in there, the oxygen would be being consumed, but it would be being replaced by carbon dioxide, and the bubble, the colored dye, wouldn't move in the tube at all. Because as the oxygen is consumed, CO2 is being released, you'd have no change in pressure. But by using the KOH to absorb the CO2, and then the oxygen being consumed by the P, you're going to change the volume of the gas, which is going to cause the pressure to change, which causes the drop to move. Did I lose you there? Whoops. I'm trying to move because time is running out. Let's, so if you look at the directions, you will see, I'm going to share it again real quick. Where did I put it? Not that one. So if you look at the directions, these are the three tubes we would set up. Here's the one with the germinating seeds right here. You can see they're a little bit more swollen, a little bit bigger. They fill up most of the container. So down here is the cotton with the KOH. Here's the non-absorbent cotton to protect the peas. Here's the germinating peas. That's all that's in this one. Here's the second setup that's got the non-germinating peas. They have smaller volumes, so you have to create the same value, you add some glass beads. So this is a mix of non-germinating peas and glass beads. And then you have one control that has nothing but glass beads of the same volume. Yay. And then you can see here are your setups. So first time through around room temperature, you would have ran the three containers. We would have then reset the same three containers and ran them around 10 degrees. So I think I misspoke. I think I said 37 for the warm. Looks like we used room temperature. which is about 23. Okay. And so here's what the setups would look like. In this case, there's a weight on them, so they would stay in the water. They wouldn't float on us. The device we were using, we didn't need the washers. Some of the setups, they'll lay them in there. Again, ours is standing upright, but you get the same results. And here's what I was talking about. Here's that air bubble. And as the seeds are using up the oxygen for cell respiration, that air bubble is going to move up the tube. And as the air bubble moves up the tube, the dye is going to move up the tube, and it's going to allow you to measure. It's going to allow you to measure how much oxygen is being consumed. Right. I would suggest doing a skim through of the reading of the original instructions. That should help you as well. How much time do I got? I don't think I've got enough. All right. So I think if you read the lab, based on the little bit I was able to show you, I think you can get the idea of what was done. I'm going to put up a link to a short video from Bozeman, where he also shows the lab setup a little bit more visually than I could. So that might also be of help to you. I just want you to have an idea how the data was collected, so you have some way to know how to process it. Right? So one setup has live germinating pea seeds, fully active. The other setup has live seeds that are dormant and not active. And the other setup simply has glass beads. First time we ran them at room temperature. Second time we ran them at cold temperatures. And so you'll see if you look at the data tables, those are the 4 data tables that are provided. And so you're trying to answer the question, how does temperature affect the rate of respiration? And you're trying to answer the question, is there a difference in the respiratory rates of germinating and non-germinating peas? Really is sort of what you're going after. Questions? I will put the link up to that short video. It might help. I'm a little leery on the video because he ends up going a little bit into some of the answer, if you would, and I want you to figure it out yourself. So I'll probably download the video and cut the end of the video off. Because I want you to do the thinking. Anyway, anything. So you got 2 things to do before coming back on Friday. You have this lab analysis, and then you have the POGIL, which is about the final step. On Friday, we'll talk about the final step. We'll talk about the lab results you came up with, and we'll probably demo the second lab we normally do for this section and start wrapping up by Tuesday of next week so that you can take the unit test on Thursday. Terpish. All right, we've hit our time, so thank you for your attention. Thank you for being present. We will see everybody on Friday. Peace out.